to each other telepathically, program new experiences directly into our brains, and maybe even live to 150. Technology is pushing from every direction, getting faster with each passing second. Prepare yourself. The future is closer than you think. The human being of the future will be a technological wonder with abilities once reserved for science fiction. Our brains and bodies will do things we've only dreamed of. Even how we speak to one another will be radically different.
I'm Chuck Jorgensen, and I'm talking to you right now directly from my brain.
Imagine a two-way conversation without moving your lips.
Are you all right?
Yes, I'm doing fine.
It's a practical form of telepathy called subvocal speech, and it's being developed by Chuck Jorgensen at NASA's Ames Research Laboratories.
When we perform the task of speaking to somebody, what happens is our brain sends a signal to the muscles that control our vocal cords and our tongue. Those signals themselves provide information, and they correspond to the words that we hear auditorily.
Our brains send us these signals even when we keep our mouths shut. And Dr. Jorgensen has figured out a way to retrieve them.
In order to capture subvocal speech, the first thing that we have to do is to catch the tiny electronic signals that will be going from Rebecca's mind to her vocal system. These are called electromyographic signals.
The signals are picked up by electrodes planted on the skin.
The electrodes basically look like this. They're tiny little pads, much like you have in a doctor's office. So if she said the future out loud,
The future.
we have the signal that's corresponding to that muscular activity. But with subvocal speech, she doesn't have to move her mouth. She could say that word silently. So she would say the future silently. Here she said the future, but she didn't move her lips. And you can see that there is still the same signal being picked up by the electrodes underneath her throat.
Once the electrodes capture the signal, they can be transmitted, as if through a cell phone, to someone with an earpiece receiver.
So the newer sensors are the size of a dime, and they're going to continue to get smaller. In the not very distant future, we see them being embedded in clothing as a matrix of sensors.
Eventually, the technology will be virtually invisible. Two people equipped with the same sending and receiving devices will literally be able to talk to each other without moving their lips. And that kind of covert conversation could change everything we know about communicating.
What should I say?
Don't answer that question.
There's many situations where saying something out loud isn't necessarily optimal.
Wait for my go. Wait for my go.
This techno-telepathy can be used by rescue workers or police to silently coordinate their movements.
Hang back, hang back.
But more practically, you could make a silent phone call during a meeting.
What time is dinner?
Or tell your date at a boring dinner party
Let's go home.
without insulting your host. But like most new technology, it could also be exploited. Students could use it to share answers on a test.
The answer is C.
Or poker players to gain unseen advantage.
He's bluffing.
However subvocal speech is used, the future of this silent communication is closer than you think. And it's not only fellow humans who will hear our thoughts.
We can actually move a robot around using subvocal commands. For example,
Move forward
Machines will also be able to respond.
and move left. Good boy.
In the near future, we'll communicate silently with subvocal speech, but there's a limit to how much our brains can process. Or is there? The future will change the way our brains work. Can technology really make us smarter? The human being of the future will be a lot smarter, thanks to pioneering research being conducted on how our brains work and how they can work better. Rather than receiving information and entertainment from a computer screen, imagine having access to information right on the surface of your eye.
To the best of my knowledge, nobody has attempted to construct images right on the surface of the cornea. It's unprecedented.
If engineers could integrate a tiny display into a contact lens, we'd become our own walking computer screen in theater. Sound far-fetched? In the future, it will happen. Thanks to the pioneering work done by Babak Parvez at the University of Washington.
This is our prototype contact lens with embedded circuit connections.
The microscopic circuits will generate high-resolution images right onto the wearer's eye.
So what the user would see out of a display like that would be an image suspended in the air, and then you could superimpose that onto the outside world.
You'll have the faces of everyone you know right on your eye. You'll never forget a name. And when driving, you'll never get lost.
Directions from your GPS unit relayed directly onto a contact lens.
These specialized lenses will revolutionize medicine, giving doctors vital patient information, even in the midst of an operation.
Imagine a surgeon in an operating room. You could superimpose the information that's already collected by other sensors right in front of the surgeon's eye. So these are borderline science fiction applications.
So why has no one developed computerized contacts before? Because the challenge is combining microcircuits with a fragile plastic lens.
Fabricating high-performance silicon circuits requires extremely high temperatures. A contact lens will never survive.
To solve this problem, Parvez's team developed a technique that allowed the microcircuits to self-assemble onto the plastic lens. The surface of the lens is dimpled with tiny receptor sites. When the team pours the pieces of circuitry over it, each tiny piece finds its correct place on the lens. Then the pieces lock together to form actual microcircuits.
Once assembled, the circuits function pretty well.
There's still the question of whether or not you'd stick something like this in your eye.
As far as the surface of the eye is concerned, there is no difference between the normal contact lens and the contact lens that we are building to incorporate all these functions.
These devices will be as easy to use as today's contacts, but it's their miniature electronic components that distinguish them from any corrective lens. These lenses of the future will even connect to the internet. Imagine surfing the web in secret from the eyeball-sized screens only you can see.
You'll have a contact lens with a wireless link not decades from now, in a much, much shorter time frame.
Well, the brain is an extraordinary object. In fact, without question, the most complicated object that humans have thus far encountered in this universe. It's almost impossible to put a limit on what its capabilities might be.
Professor Gary Lynch is a pioneer in neuroscience research at the University of California at Irvine.
We're working on ways of enhancing the capabilities of the human brain.
That work involves not only trying to improve the brain we have, but keeping it at maximum potential throughout our lives.
The trouble with the brain is that it begins to fail with age.
Today's human brain power peaks at the age of 20. Then it begins a steady decline.
Why? Why should this thing spend 18 years to develop, to wire itself up, to form these thousands of trillions of synapses, and then start to decay almost as soon as it was finished?
It's a problem this 60-year-old PhD is trying to solve.
The fact that a third-year graduate student has more mental agility than I do is a simple fact of science, and it's one that me off.
Lynch is developing a drug that would keep our brains from deteriorating and even make them work better.
These new class of drugs, which are called ampakines, gives the brain a new kind of power.
Our brains are made of neurons, more than 100 billion of them.
Thought is nothing more than a community of neurons talking to each other for a fraction of a second. Each neuron passes its message on to the next. Not bad, not great.
What would happen if you could accelerate that process?
With ampakines, the jolt of electricity is larger. More neurons are going to respond to the message, and more neurons means that they'll recruit more neurons. Now you have more neurons working on a single thought, and they're a lot faster.
Our neurons send signals across tiny gaps called synapses. Dr. Lynch can measure just how much ampakines grease these synapses inside the brain.
What we have in this dish is a piece of the living brain. And up here on this screen, you can actually see the synaptic voltages. You can hear neurons receiving their messages and preparing to pass them on to the next neuron. And now, let's add some ampakines. Same synapses, more voltage.
By increasing the speed of thought, these drugs will make us more alert and improve our memory. But they'll have none of the jittery side effects of stimulants like energy drinks or coffee.
Ampakines have been tested in 65-year-old humans and shown to improve memory quite substantially without any evident side effects.
Smart drugs will become part of our lives as we fight off brain degeneration.
Ampakines are not a speculation. These are real things that are now in testing by pharmaceutical companies. Potential is there to do things the brain cannot now do. And that opens up a new book in the human story. What comes next is going to be really, really fascinating.
Right now, it's predicted that by the year 2040, more than 100 million people worldwide will suffer dementia. That's 1 in every 15 adults over the age of 60. A drug that can slow the loss of memory will be a breakthrough in the fight against Alzheimer's, strokes, even schizophrenia. And for those who remain healthy, a vigorous, youthful brain will make life more enjoyable. Ampakines will give the brain more speed, add more processing power, and you've got a brain that's superhuman.
Humans have a very, very limited capacity to add new neurons into the brain after birth.
Dr. Carlos Lois is working on a way to add more neurons into the brain. It's called neurogenesis.
If we figure out a way of getting new neurons added into the circuits of adult brains, that would mean as adults we could learn new things in the same way that children learn them.
Hello. Bonjour. Konnichiwa.
To figure out how humans can add new neurons, scientists like Lois are studying songbirds. Why songbirds? It's because they can learn up to 2,000 different songs. Songbirds are able to add new neurons throughout their lives.
So, what do you think?
With more than 10 billion neurons making trillions of connections in our brains, there is a risk. Adding new neurons and more connections might interrupt existing ones, like overwriting an existing document. The possibility? Erasing certain older memories.
You add new neurons into the brain, you have the ability to learn a completely different language, you have the ability to learn how to play violin, but it's also possible that maybe you could forget who your wife was. Maybe that's not a bad thing in some cases.
Still, if Dr. Lois manages to unlock the songbird's secrets, the rewards of achieving neurogenesis would be mind-blowing. With all due respect, your superhuman brain will be smarter than your current brain is capable of understanding. Not only are scientists working to make our brains function better, they're also investigating new ways to use our brains.
The brain, what is that all about?
Everything we experience comes through our 5 senses. But Dr. Rodolfo Linas has a technology that could make your 5 senses obsolete. Someday you might be able to see even if you're blind, hear even if you're deaf, or recover memories that you thought were lost.
A new set of technologies that might allow us to talk to the brain directly. We humans are visual animals. Let me draw it for you. It is something like that. Let's put an eye here. Now, for most people, seeing is something you do with your eyes. The eye connects to the thalamus. But you know, it is really not so. The eyes don't see. It is the brain that sees.
When you dream, you see, but you're not using your eyes.
In principle, one may be able to produce seeing by activating the brain directly.
Dr. Linas plans to run wires into the part of the brain responsible for vision. The wires would stimulate specific brain cells with electrical impulses, creating images in your mind. You'll see, even if your eyes are shut.
You can have sensations, you can have images as richly as you want to.
Linas believes he can also hotwire the parts of the brain connected to hearing or touch. This technology paves the way for true out-of-body experiences. You'll be able to see the world without ever leaving home.
What if we want to travel? Why don't you just plug it in directly? And then you will see it, and then you will feel the breeze.
If your brain can experience sights, sounds, smells, and sensations of being somewhere else, could virtual travel be as good as actually being there? Could armchair thrill seekers skydive in complete safety? Or even walk on other planets? Dr. Linas believes there's no limit to the images and sensations that can be sent directly to the brain. How will all of this information get there?
Through the vascular system.
The brain is filled with tiny blood vessels that deliver oxygen to each and every neuron. Dr. Linas plans to run bundles of microscopic lines called nanowires right through these blood vessels. They will carry the signals containing the sights, sounds, and other sensations to the appropriate parts of the brain. In the future, Linas also envisions being able to hardwire our brains directly to someone else's.
You will have not only the ability to address sensation, but also emotions.
You won't need words to communicate your feelings. The person on the other end will be literally inside your head, feeling your love or anger. Physicians will no longer ask, "Where does it hurt?" They'll know.
We're beginning to get the tools to interact directly with our mind.
But whatever our brain's potential, we'll still want a healthy body. And another nano-based technology might make it possible to rebuild broken bodies and make strong bodies even stronger.
We do not want to alter the human body. We just want to improve it.
Dr. Sam Stoop likes to build and fix things, but he doesn't use arc welders or screw guns. He uses nanotechnology to regenerate parts of the human body. Nanotechnology refers to the building of objects and machines so microscopic that the building blocks are actual molecules, which are even smaller than cells. They're called nanofibers.
Our favorite element in the toolbox is a nanofiber. Structures that look literally like a fiber, except that they're extremely small. Suppose you had a hair, human hair. I don't have any left, but imagine that you could have 100,000 split ends in that human hair. Each would be the size of our nanofibers.
It's their size that makes these infinitesimal objects so promising to the process of human reconstruction. They operate at the cellular level.
We are creating a toolbox of nanostructures to regenerate parts of the human body.
These nanofibers could be applied to an injured kidney or heart. The fibers will connect to the weakened areas inside the organs, creating miniature bridges between the cells. The fibers are then able to signal the cells to divide and multiply over the damaged area, enabling the body to repair itself.
The nanostructures talk to the cells, tell cells to proliferate, to divide, so that something can grow.
This nano process of regeneration can be applied to almost any cell structure in the body.
For example, if you could regenerate insulin-producing cells, then you would be curing diabetes. In Alzheimer's disease, it is important to have the ability to regenerate neurons.
But Dr. Stoop doesn't just want to fix an injured body. He hopes to make every human body stronger.
The objective of regenerative medicine is to improve the human design that evolution has given us. For example, cartilage does not regenerate anymore after you turn 18.
Nanotechnology will be used to rebuild your cartilage, creating stronger joints. Athletes could prolong their careers, and everyone will be able to stay active well into their 90s.
Regeneration is about improving quality of life. It may allow us to live a longer life as well.
We know that some animals can regrow lost limbs. If they can do it, why can't we? In the future, we just might. The future may hold the solution to one of humankind's most traumatic experiences, the loss of a body part. Scientists are exploring a number of different ways to actually regrow them.
My name is Ellen Heber-Katz, and I believe that this mouse is the future of regeneration.
Dr. Heber-Katz happened upon the unique abilities of her mouse by accident. She was studying mice that were missing part of their immune system.
We have been looking at a drug that we thought could inhibit autoimmunity, and we ear-punched the mice so we could distinguish the treated from untreated. We injected the mice with these various compounds. 3 weeks later, there were no ear holes, so I couldn't tell which mouse was which. If this was real, this would be incredible. I mean, this was really super healing.
Ear punches don't heal by themselves. And that's not all. Dr. Heber-Katz discovered that when they cut off the tails grew back. Same with the toes.
In these mice, you can make an injury to the heart and the tissue regrows without scarring, normal architecture and normal function. If we can determine what the exact properties are of this mouse, what is the biochemistry, we could then apply that to humans and regrow organs, regrow limbs, regrow digits. The possibilities are really unlimited.
Humans have always had a limited ability to regenerate.
You certainly grow hair, you grow nails. That's more cellular regeneration and tissue regeneration.
But not much more, certainly not limbs. It's only amphibians like salamanders and newts who are able to regrow tails, arms, or legs. That evolutionary ability has been lost in mammals. But Heber-Katz's mice seem to have regained it. The reason may have to do with their suppressed immune systems. The immune system is what protects us against disease. One way it works is by scarring.
You have a wound and you create a significant scar, and that blocks bleeding. It blocks infection, but it also blocks growth.
Forming scars might prevent our natural ability to regenerate. Check out this microscopic view of a typical wound, healing. And then watch how the cells of Dr. Heber-Katz's mice do the same job.
The cells are phenomenal. They move about 10 times faster than normal cells.
The implications of Heber-Katz's discovery are extraordinary. If mammals like these mice have the hidden power of regeneration, then scientists may learn how to unlock this ability in humans as well. Imagine the difference that could make to people who have lost their limbs.
I think it's looking very promising to regrow digits and hopefully in the future, whole limbs. The ability is there, but we have to turn it on.
Will the ability to regenerate create a world of daredevils who approach life with reckless abandon, knowing that if they lose a limb, it will grow back? Will we all be braver in this brave new world?
It's interesting because regenerating species are often considered to be immortal. We could increase health, we could increase longevity. I think that there are incredible possibilities in the very near future. It's not a question of if, it's a question of when.
If the prospect of regrowing limbs sounds amazing, what about our internal organs? Right now, the only way people get new organs is through transplants. The wait is sometimes years.
My name is Gábor Forgács. In the future, we'll be able to harness nature's ability to form organs and build our own.
Dr. Forgács and his lab are pioneering a radical way of building new internal organs. They're creating the organs cell by cell, literally printing copies of the original organs nature gave us.
Organ printing sounds really like the ultimate science fiction.
This technology is considered the future of tissue engineering. It's based on the fact that a cluster of human cells, what Dr. Forgács calls bio-ink, behaves just like a liquid. When placed next to one another, the clumps will flow together and fuse, forming layers or other shapes.
These are the bio-ink particles. Each of them is composed of about 10 to 30,000 cells.
To build 3D structures like organs, Forgács and his team use a printer to assemble the human cells into the desired shape. It's similar to how an inkjet printer produces words. Cells survive the printing process well.
This is the bioprinter.
Your printer can't assemble solid objects, let alone a human organ. But this one will. The printer creates a sheet of cell-friendly biopaper. Then it prints out the living cell clusters onto the paper, drop by drop. Once in place, nature takes over. The cell clusters fuse to each other, creating more complex tissue structures.
We cannot control every detail of the process, but it turns out that this is not necessary because the biological material, by its fundamental nature, is able to self-organize.
As each layer is completed, another layer of cell clusters is added.
We build the first story, then we build the second story, and we build the third story. And that's how we get to the skyscraper.
Eventually, the biopaper separating the layers dissolves, and the individual layers of cells fuse together.
And that's the way we build three-dimensional structures.
Since cells from any organ can be put through this printing process, Forgács foresees using this technique to replace any damaged organ. And since the organ would be printed from your cells, there would be little chance of rejection.
Imagine someone who had bypass surgery. That person is living with a heart which is not fully functional. So I can envision printing sheets of cardiac tissue we can attach to the damaged heart. Will I be able to replace my liver because I'm a heavy drinker? Will I be able to replace my lungs because I'm a heavy smoker? Well, my first advice is don't be a heavy smoker. But having said that, of course, it is quite possible this technology will be useful within one's lifetime. Why can't we imagine that at some point when you need to replace a damaged organ, you are rolled out of the operating theater with the new organ that was printed into your body? I think that what was science fiction 50 years ago is close to reality today.
In the future, we'll be smarter, stronger, and be able to order in new limbs and organs. You'll also be able to predict just how healthy you'll be, or which disease has your name on it.
Understanding the genetic code, understanding our genes, is changing the entire basis of medicine.
Craig Venter is not just a pioneering geneticist. He's also the experiment, the first human being to put all 6 billion letters of his genome in sequence.
If we printed out my genome at a reasonable font size so you could read it, it'd be a stack of papers higher than the Washington Monument.
And it forced him to face some inconvenient truths.
I found in my own genetic code I have a risk not only for heart disease, but for Alzheimer's disease.
With the genome mapped out, scientists are now making breakthroughs every day about how our DNA plays a role in our overall health.
We'll never get the information that'll say at age 62 you'll die from colon cancer. We'll have the information that says from the genetic code you maybe have a 30% increased risk of getting colon cancer over your lifetime. Only by understanding large populations will we know what those probabilities really mean.
One day soon you may be able to predict the future, at least when it comes to your health. Pioneering work on genes has the potential to tell us what diseases we face, what we could die from, even the risks to our unborn children.
The DNA that makes up the genome contains the information that goes into making all life on Earth.
The genome contains the biological information that distinguishes each of us from the other. It may also tell us about the future, thanks to research being done in Iceland, home to some of the longest living people in the world.
My name is Kári Stefánsson, and my family has lived here for at least 1100 years.
Iceland has only 300,000 inhabitants.
It is extraordinarily convenient because we can study everyone in Iceland with a particular disease, so we don't select from a group of patients, we just take them all.
Dr. Stephenson hopes to identify the genetic causes of disease. He has embarked on a quest, asking the entire population of Iceland to pool their biological testimonies to advance scientific progress.
The importance of the sequence of the DNA of an individual only becomes meaningful when you look at it in the context of the DNA of other individuals.
Deep in the basement freezers of Decode Genetics are the actual DNA samples of more than 100,000 Icelanders.
So I can take anyone in Iceland and I can basically tell you how he is related to everyone else in the country. What we are interested in is comparing the genomes of many people.
People will have a profile of their genome in 5 to 10 years, just like they know the blood groups today.
With that knowledge, we will know what the future holds.
My high-functioning LAC1 gene means I should live up to 90 or beyond.
My gene, AMPD1, is in good shape, so I don't tire easily when working out.
Not only does the genome dictate the color of our hair, the color of our eyes, etc., but it also decides how we respond to our environment.
Because of my extra long dopamine receptor 4 gene, I like taking risks.
How we respond to the food we eat.
All I know is that my genes allows me to drink as much coffee as I want.
Your genome also holds the secrets to your future health.
I'm convinced that the discovery of variations in the sequence of the human genome is going to be the foundation of the development of new preventive medicine.
New tests for disease-carrying genes will identify problems before they arise.
We have the test for type 2 diabetes, which is now becoming an epidemic in the United States. We have a test on the risk of early heart attack.
For Icelanders, this coming genetic insight will give them power over their future.
It's definitely going to empower you. It allows you to seize control of your own health care. I have very little genetic risk of Alzheimer's disease. I have relatively high risk of prostate cancer. I have relatively high risk of type 2 diabetes.
So I'm sort of an average person when it comes to genetic risk of a disease. And there is nothing more depressing than being an average person, I can tell you that.
In the future, your date might casually inquire as to your genetic profile. After all, most people choose their life partners based on what we now know are largely genetic characteristics.
In our selection of a mate that looks in a particular way, we are being driven both by the genetics of the person we are driven to and by our own genetics.
But what happens when we are able to read the blueprints of our unborn child? Will those with the means be able to create their own genetically superior children?
It will be possible. There's little question about that. We can do a lot of that already today. What is the wisdom of doing that? I think it could be actually somewhat dangerous to human survival if we would begin to manipulate these common traits on a large scale. You are making fundamental decisions for the generations to come. I think that people should always have the right not to know when it comes to themselves. But the person who doesn't want to know is not going to prevent the world from making discoveries.
Our genes may hold the answers to our future health and what we can expect as we get older. But what if we never get older? Is it possible to actually stop the clock?
You can imagine someday a small pill being taken once a day, or even less frequently, to treat diseases of aging.
Mapping the genome will give us the answers to the future of our health. And how we manage our health is going to change our lives. But it's only one part of a bigger change: human life expectancy. A century ago, the average lifespan was 60. It's now up to 83. But could the process of aging actually be stopped? New aging research may just lead us to the legendary fountain of youth.
In the future, we'll be able to rejuvenate human tissues.
Dr. Howard Chang thinks he's found a way to fight aging. And he's not just talking about how you look.
It's absolutely not an attempt to make the new Botox, not a new attempt to get rid of wrinkles. The focus of our work is really to understand, why do cells get old?
Chang's research focuses on the skin.
The skin is a quite special organ. The human skin actually turns over once every 28 days. All the cells that used to be there are replaced by new cells. So every month you look in the mirror and it's a new person that you're looking at.
The unusual thing is that in an old person, these newly born skin cells still behave as if they're old. Which means aging is not simply a matter of the body's cells wearing out over time. It's a function of our genes.
So if there's actually an active genetic program that's making cells old, then it might be possible to turn off this program to return to old cells back to a youthful state.
Every cell in our bodies has a protein called NF-kappa B. This protein turns various genes on and off like a switch, including those responsible for aging. Dr. Chang was able to turn off this master switch in an old mouse.
We engineer a mouse to carry an extra gene so that we can turn the aging gene off by applying an innocuous chemical to the skin of the mouse. So this mouse has basically had a normal life, and when it's already quite old, we can at will deactivate the aging gene and ask whether the mouse may now acquire some youthful characteristics again.
The test results are astonishing.
While the rest of the mouse is old, just a patch of the skin that we treated became young again.
The same results may occur in other parts of our bodies besides our skin.
We believe that the same genetic process, that is the gene program of aging driven by NF-kappa B, is actually active in many other tissues, including the brain, the heart, the liver. By blocking the genetic program of aging in different tissues, one might be able to generate healthful benefits for age-related diseases.
It's possible that by silencing our aging genes, we may never grow old.
I think it's very possible that down the line, in the future, we could actually extend lifespan.
Dr. Chang's discovery won't make us teenagers again, but it might keep us healthier for many extra years. Manipulating our genes is not the only way we could live longer. Other scientists believe all we would have to do is pop a pill. The research behind this anti-aging wonder drug is based on the phenomenon called calorie restriction, otherwise known as a starvation diet. Though how it works is still a mystery, it's believed that when an organism is starved, this triggers a gene that strengthens its cells. The cells end up living longer.
For 70 years, people have known that calorie restriction extends the lifespan of many organisms. And what was needed was a molecule, a small molecule you could put in a pill and just take that once a day and have the same benefit.
Doctors David Sinclair and Christoph Westphal think they've found that molecule.
It's called resveratrol. It's found in red wine. Well, resveratrol is a remarkable molecule in itself. It has extended the lifespan of every organism we've tested. It extended the lifespan of these yeast cells about 30%. And it's been tried on worms and flies, and they also live about 30 to 40% longer. Fish live 60% longer.
Resveratrol has the same effect as calorie restriction. It stimulates the growth of an enzyme called SIRT1 that helps to regulate our cells.
SIRT1 is a master controller of the cell's defenses, like the 911 call center, sending out the defenses like ambulance, go out and do that, fire brigade, go out and do that. Instead of normally it's saying, repair DNA when you feel like it, protect the cell when you feel like it, along comes resveratrol, binds to the protein and says, it's urgent, let's protect the cell, do that, do that. What we get as a result is a healthier, longer-lived animal.
But could it work for people?
The good news is it's worked on so far every organism we've tested. Just recently we fed mice resveratrol, and what we found is that the mice are much healthier. As they get older, they don't show the signs of aging. We can extend the lifespan of mice by twofold. They could run further, they were leaner, they didn't get heart disease, they didn't get diabetes. What we've stumbled upon is really the body's natural defenses against disease and deterioration. We've now made molecules that are synthetic, but are 1,000 times more potent than resveratrol.
The longevity pill, a drug to cure old age. In your lifetime?
We're talking potentially about drugs that can target the genes that control the aging process within the next 5 to 7 years. Our goal, of course, is to improve healthy lifespan. And that would mean we would be more productive for a longer time.
Thanks to the daring work of today's scientists, life in the future promises to be radically different than it is today. We will be self-healing, disease-free, smarter, genetically superior. We will live longer, a lot longer. The revolution in genetics, molecular biology, nanotechnology, and microchip informatics could create a world of superhumans. We may not have to imagine such a world. It may soon be ours.